Okay. Can you all hear me online? Box is green, so that's good.
CN is online. Are you able to share your screen?
Yeah, I'm trying to figure out how to do that. Let's see.
Yep. There you go. Why don't we send on the chat if they can ask if they can hear us?
Okay. And somebody on Zoom pop on and give a thumbs up if you can hear me.
Yes,
Oh,
we can hear
okay.
you and we can see the slides.
Awesome. All right.
Wait 1 minute. See. You want to turn your camera on?
Sure.
So I think what's happening is we're not getting the owl. That's my guess.
We can see you and we can also see the slides and we can hear you.
Awesome.
Okay.
Yeah,
Yep.
I think so.
I think we can get started. Welcome, everybody. This is our second last seminar of the semester, and it's my pleasure to introduce Craig C., who is researcher level 6 in the Department of Agronomy and Plant Genetics. Craig did his PhD here in ecology, evolution, and biology with Dr. Sarah Hobby. And so today he's going to tell us a little bit about nitrogen, its mobility, phosphorus. He says it's set in stone, and soil nitrogen stocks are driven by phosphorus availability in terrestrial ecosystems. So thank you, Craig.
All right. Thanks, Tim. So yeah, I guess today I'm kind of recycling a talk I gave at the Soil Science Society meeting this year, which is sort of a summary of my work on slowly cycling phosphorus, or what I call slow P. Slow P could also stand for slowly cycling projects or slowly cycling papers, because what I'm presenting is like a bunch of unfunded side projects that had nothing to do with my PhD work or my postdoc. But I've been chipping away at it for like a decade. So this is soil, water, climate. I probably don't need to convince anyone here about the urgency of the current carbon dilemma. We've increased atmospheric CO2 levels by about 50% since the Industrial Revolution. We know we need to stop burning fossil fuels. The good news is that we actually have a pretty good handle on quantifying the scale of the problem, right? We have pretty good estimates of how much carbon we're adding to the atmosphere each year because there's dollar signs attached to it. We measure it in barrels of oil, tons of coal, etc. Ironically, we're not nearly as good at quantifying the way carbon is cycling through our natural systems, which is actually just as important for predicting the future. Case in point, this figure is from a recent paper that applied an ensemble of some of our best upscaling models across the Arctic and Boreal region. So that's about 15% of the land area and stores about a third of the world's carbon, arguably the most vulnerable carbon or soil carbon. So the x-axis here is net ecosystem exchange predicted by the model ensemble, and the y-axis is net ecosystem exchange from the exact same location, actually measured on the ground using eddy covariance. And as you can see, the fit is not great. The R is 0.27, so the R squared would be about 0.07. So this tells us that we definitely need a better understanding of the site level drivers of the carbon cycle. So what can decades of ecosystem ecology contribute to the conversation? Well, we know that nutrients, specifically nitrogen and phosphorus, drive primary productivity. So this meta-analysis is almost 20 years old now, but I think it's probably one of the more important figures in a paper in modern ecology, because to me, it sort of puts to bed Liebig's law of minimum in natural systems. So remember Liebig's law, anyone? Raise your hand, no? It's basically an extension of the concept of the limiting reagent in chemistry, right? So it's the idea that ultimately there's one limiting nutrient in a system. Usually it's nitrogen, right? Yeah, Sarah Hobby teaches the concept with the analogy of peanut butter and jelly sandwiches. So if you're making a bunch of PB&Js and you run out of jelly first, you can't make any more sandwiches till you get more jelly, right? But I would actually argue that most people would just like skimp on the jelly, use the rest of the bread up. You know, you got like crappy sandwiches, but you make it work, right? And this figure is, I think, kind of shows that. It's the response ratio of fertilization experiments, NP and M plus P, across different ecosystems. And so if Liebig's law were strictly true, right, if nitrogen was the only limiting reagent in grasslands, you wouldn't see a phosphorus response at all. But we do pretty consistently, right? And in forests, if phosphorus was the only limiting nutrient, right, you wouldn't see a nitrogen response, but we do. And when you add the two together, you get this kind of synergistic, non-additive effect, oftentimes, which is pretty cool. Yeah, so N and P together kind of drive carbon inputs to most terrestrial systems, or at least they're important. And they also drive carbon losses via decomposition, at least in litter, right? So these are standardized within site slopes for the relationship between litter traits, different litter traits, and then decomposition rates, the K value. And on the left, it's leaf litter it's a leaf litter meta-analysis, and on the right, it's a root litter meta-analysis. And this shows basically that higher NMP concentrations lead to faster decomposition pretty consistently across sites. So globally, NMP are important controls over ecosystem carbon cycling, right? And the modelers know this, right? There's a growing consensus that N and P are going to be major constraints over future carbon gains across the world, right? And we need a better understanding that changes in nutrient availability are going to be critical to then predicting the carbon dynamics. The problem is that the processes controlling NMP dynamics are like super complex, and they're full of feedbacks, both positive and negative, and the data are sparse. So it's a tough nut to crack. And I guess that's kind of a question that centered my research for the interest for the last 10 years is what are the feedbacks between nutrient limitation and nutrient cycling? And so I've jumped around topics and ecosystems a lot, but this is sort of a common thread. And I first got interested in the topic during my master's work at the Hubbard Brook Experimental Forest in New Hampshire with Dr. Ruth Yanai. So this is a long-term ecological research site, for now still funded by the National Science Foundation. LTER is the acronym. I'm going to use that a lot. Yeah, here I kind of helped set up a long-term NNP forest fertilization experiment, replicated across 13 stands. As part of that, I was tasked with doing all the pre-treatment leaf sampling, so I was really into foliar chemistry. And so for those of you who haven't sampled leaves in mature forests, the standard protocol involves a 12-gauge shotgun and steel bird shot, because you got to get representative canopy samples, right? So I went around shooting hundreds of trees and then came back to the same places in the fall, got my leaf litter samples, digested everything and ran it on ICP. That's me doing science stuff. Note the hair, it was a long time ago. And so I was really at the time interested in foliar resorption, which is the percentage of N and P that leaves are sucking back up into their stem tissue before they drop their leaves in the fall, right? So when leaves are changing colors, they're senescing, they're taking nutrients back up, and it's like half or more of their nutrient demand for the next year. So it's important, right? And so this figure was from the paper, the original paper from that work. It wasn't the main point of the paper, but it stuck with me. So the x-axis here is the ratio, the N to P ratio of fresh foliage for the entire tree canopy across 18 plots. So it's like averaged across all the tree species and Daisleria and everything. And the y-axis here is like a ratio of two ratios, so I apologize. The y-axis, it's the ratio of N resorption to P resorption. So the plots below the one line here, the canopy is conserving proportionally more nitrogen than it is phosphorus. And if it's above the one, it's conserving proportionally more phosphorus than nitrogen. And what's cool is that these resorption ratios lined up really well with published thresholds for nutrient limitation at the time. So the N to P, foliar N to P ratio on the X, people often wave their hands and say that's things are N limited or P limited, depending on where it is. So this was cool, I thought. It's also like a nightmare for a master's student who just took like regression analysis because like the spread of the data is pretty ugly, right? You got like this cluster down here and it almost looks like it's positive, and then you got these 3 points, right? And it's cool because they fall right in that range, but it's like, what do you do with that, right? It's hard to say if it's real. But luckily, a few years earlier, a bunch of people dug a bunch of really big holes in all of these plots. And it turned out, looking at the quantitative soil pits data from all these plots, that there was like a ton of primary mineral phosphorus in these 3 plots. And so it totally like validated that they should be nitrogen limited, right? So that was, I thought, validating, and it really kind of impressed on me the importance of thinking about these like so deeper kind of slower cycling P pools on ecosystem processes, because it basically predicted N limitation. So after my master's, I moved down south for a year and took a job as a data analyst at the Coweta Hydrologic Lab in Western North Carolina, which is also an LTER site. And there I was kind of tasked with helping synthesize changes in 20 years of carbon and nitrogen pools that Dr. Jennifer Knipp had been measuring since the 90s. There's Jennifer standing on the platform. So this was 5 forest stands that represent the major forest types of the region. And they've been pretty intensely monitored as part of the LTER program. Note though, all CNN, they really weren't, there was P stuff looked at, but it was like afterthoughts, wasn't part of the grant. So here's the figures from that paper. This is the change in AMPP measured every 2 years using tree surveys by Jim Clark's group at Duke. As you can see, productivity, carbon uptake, right, increasing through time. And this shows annual litter fall nitrogen flux. And this is actually one of the best long-term litter fall data sets in existence, I think. Like, it's like it's like classic Forest Service, like they grabbed it like 10 times a year and compiled like, so yeah, basically N flux is increasing too. So we could say pretty conclusively that things are changing, right? Accelerated CNN cycling, presumably due to increasing CO2 and long-term cumulative nitrogen deposition, right? And like I said, these plots represent a big environmental gradient of N deposition. So we were actually kind of surprised that differences in N deposition didn't really explain like the rates of change. Like that's where we were going with it, and we just couldn't find any evidence. We looked. And so we published these figures, and then around that time, we submitted the paper. I had started a PhD here in mycorrhizal fungi, And then I found out that there was phosphorus data from these plots, and I was like, oh, I need to pull this together, you know? But like I said, P wasn't included in the grants. The measurements were less consistent. But what you're looking at here are the means of malic phosphorus extractions that were taken at the beginning of the time series, like in the early 1990s, and then at the end of the, or towards the end of the time series. And as you can see, available phosphorus like shot up. It more than doubled at all of these sites, right? And side note, the first, there was no change in pH during this time. That was like the first thing I looked at, and they did pH on all of these extractions. Like there's no pH wasn't changing, but available P went up. So wouldn't it be cool if these changes in P corresponded to the changes in carbon and nitrogen that we observed? Whoa, they do, right? So the x-axis here now is that increase in available P. So it's the difference between the two bars on that left figure, right? And the y-axis here is the increase in nitrogen flux. So literally just the slope from that regression I showed you in the previous slide. And here's the same, but with the y-axis is now the increase in AMPP. So this is super cool, right? Empirical, like long-term empirically observed changes in NP and C cycling, and they're all synchronized together like pretty well, just like you'd expect in like a model or something. But we can't really say what's driving what here, right? You could actually, and I think anyone could sit down and if they wanted to make a good argument for putting any of those three variables on the x-axis, you know? More N deposition leads to greater phosphatase in soils. So maybe the N's on the X and it just increased the P, right? So that's not super satisfying. But here's where it gets really cool. In 2008, Jackie Mohan's student at UGA, Caitlin, did a full phosphorus fractionation on the soils from these plots. And these are totally different samples from the malic data I just showed you in those bar graphs. But so the x-axis here now is primary mineral phosphorus or apatite, right? Which cycles like on geologic time versus the change in the malic phosphorus. So that change in P availability. And like, I don't know, I've never gotten a line like that before in my life. And that's like not something I would have, I don't know, I didn't expect it to work that well. So it's really cool. And it also, that apatite pool also predicted the increase in nitrogen flux, like the slope of the line for the nitrogen flux through time, and the increase in productivity. And again, this is cool because apatite has to be on the x-axis here, right? There's no conceivable way that like a little extra litter fall nitrogen changed the mineralogy of these soils. Like this is set in stone, right? So we can start to infer causality. So I was like super pumped about this. By the time I figured it out, or I guess in conclusion, we saw decadal changes in the rates of carbon and nitrogen cycling being constrained by phosphorus availability, presumably coming from these slower cycling fractions. And so right around the time this was sort of getting figured out, I was starting a postdoc at Northern Arizona University, where I met Ellery Vaughn, who was doing her master's thesis on N&P limitation at the Bonanza Creek LTER, which is up in Alaska. And so Ellery was interested in really similar questions, and we realized that between the two of us, we actually had data from 3 LTER sites. We had Hubbard Brook in New Hampshire, we had Coweta in North Carolina, and then we have Alaska. So we put in for a workshop at the LTER All Scientists meeting to try to kind of find other sites and just get some smart people together to put their heads together. And the workshop, I would say, was a success. We met a bunch of awesome collaborators. We came up with a conceptual model, which I'm going to try to share now in story form. So let's just, let's say you're feeling a little stressed out, right? Like you watch the news or something. And it's not great, And you're a proactive person though, so you decide you're gonna purchase a Sphere of Tranquility terrarium for $79.99 from Riverwalk Floral Designs in San Antonio, Texas. This was way funnier in San Antonio, Texas. But But the sphere of tranquility works, right? Everything is serene inside of the sphere. Your blood pressure is like dropping, right? And what's even cooler is the creator of the sphere was like a huge fan of ecological stoichiometry. So nutrient wise, this is like a perfect closed system. And everything is like in exact Redfield proportions. So the flux of N and P between the plants and the soils is occurring in perfect synchrony, right? The N to P ratio of the litter matches the N to P ratio of the soil, which matches the N to P ratio of the mineralization rates, which matches the N to P ratio of plant uptake. It's just traveling together and it's great. But we know this isn't how natural systems work, right? They're unbalanced. They're not closed systems. So I'm gonna buckle up now and leave the sphere. So side note, I've always had like a really hard time finding like comparable diagrams whenever I put up NMP cycles. Like they never match like stylistically or anything. So I figured like ChatGPT is gonna like take our jobs. So like let's let it figure it out. So here's what we get for nitrogen. Which is pretty good. The demonification arrow could maybe be wider. But it's all right. Here's what we get for P. You got like fish rain happening up there. It's pretty cool. I might make t-shirts, but so our jobs are probably safe for the time being, I guess, is what I'm saying. That's one takeaway. So let's go back to how NMP cycles are represented in the ecosystem ecology textbook, Chapin textbook. Here it is, your N and your P cycle. So the first thing you'll notice is probably that the N cycle is a little more complex. This is definitely due in part to historical biases in ecology towards studying nitrogen. It's also easier to study as isotopes, they are stable isotopes. But it also reflects the fact that the N cycle is just more biological and the P cycle is a little more geochemical, right? So you're probably used to seeing it represented, P availability represented like this, something like this, like in terms of pH, right? Like sorption and all that. But this isn't really a cycle in terms of like inputs and outputs. So I do think that the Chapin comparison illustrates like a pretty important point, and that's that the N cycle just has way more inputs. You got deposition, fixation, way more outputs than the P cycle. You've got denitrification. There's way more leaching that occurs. Fire is gonna leave the phosphorus behind in the ash. It's gonna volatilize all the nitrogen. And phosphorus, in the absence of like major disturbances or like systems that have a lot of erosion, it kind of stays put in the soil. It just changes forms, at least on like successional time. If you're going geological time, there's losses. So the N cycle is more open than the P cycle. So now we can go back to the sphere of tranquility, which is now just a generic terrestrial ecosystem with the box around it. The blue arrows are represented, or N fluxes are blue arrows, P fluxes are red arrows. And we can add inputs and losses. So here's our nitrogen inputs and losses. Our phosphorus ones are much smaller, right? It's functionally almost a closed system for phosphorus. And finally, phosphorus is kind of represented, it's just largely internally cycled with new inputs kind of coming from these slower pools that are already present within the system. And if it becomes in excess, kind of in that actively cycling pool, it can kind of return there, right? But, because of the sphere, right, appreciable increases in the internal cycling of one of these, like between plants and soils, you really need an increase in cycling of the other, right? So that leads to the interesting hypothesis that the ability of phosphorus to move from slow pools into actively cycling pools could ultimately limit the rate of N accumulation. So here's two diagrams that differ in the P supply from the slowly cycling pool, but have the same amount of N inputs, right? The blue arrows on the left, same size. The lower phosphorus system loses most of that new N, so the arrow out is roughly the same size as the arrow in. Well, the higher P system is able to retain more of the nitrogen because it can match it with new P. So this was basically the diagram that came out of that LTER workshop in 2022. Also around that time, Ed Rastetter published a paper using the multiple element limitation model to predict long-term changes across a bunch of different LTER sites that were parameterized with LTER data. Some of them were present at our workshop. And the MEL model is really cool, but I'm going to spare you the details of it because it is gnarly. But I'll just give you a highlight of the paper. So based on their simulations, they found the NMP constraints on future carbon gain depend on the ecosystem's availability to accumulate more NMP, to redistribute NMP from soil to plants, and to adjust the N to P stoichiometry of plants and soils. And this sentence is straight from the conclusion sentence. Because N cycles are generally more open than P cycles, ecosystems can adjust N gains and losses to match the net gains in P. So this was super neat because the model conclusion basically matched our conceptual model from the LTER workshop. And a lot of those sites were represented at the workshop. So we kind of felt like we had momentum, and a bunch of us applied for working group funding through the National Center for Ecological Analysis and Synthesis, which hopefully is still going to be there. So I want to take a second just to acknowledge my collaborators on this, as well as NCs itself. This is really an amazing group of scientists that I've gotten to work with for the last couple of years on this. And at a bunch of different career stages, bunch of different systems. It's just been, yeah, everyone's great, and it's been a ton of fun. If you ever get a chance to participate in an NC's group, you should do it. You get to spend a week in Santa Barbara, play volleyball, there's escape rooms, there's good restaurants. This is our group checking out a soil pit that the city was digging. But you spend most of your time in a room trying to figure stuff out. It's great. I mean, they have all the resources for you, and everyone has space to just work. So we'd originally set out to recreate my COETA analysis, try to do that at a lot of sites, but we quickly realized that there wasn't going to be the data to do that properly, which is it's just a constant issue with any kind of synthesis work, right? So we decided that for things to be comparable, we'd focus just on soils, specifically soils where there were P fractionations done. So our overall hypothesis was that if the N cycle adjusts to the P cycle, then spatial variability in soil N accumulation should reflect spatial variability in total soil P, right? But in highly organic soils, right, N and P are always coupled, so you could make this relationship, and someone could argue that it's just a function of organic matter and that it's not P driving it, right? So for that reason, we kind of had our secondary hypothesis that if P is truly driving it and not vice versa, then the positive correlation should also be present when you take away the organic matter and just look at the slowly cycling inorganic fractions. So we compiled all the data sets we could from the LTER that had both total N and P from the same cores or the same soil pits, or total N and a P fractionation to get at that slow pool. And we included non-LTER sites if LTER co-PI had the data set, because we weren't going to like turn it away because there just weren't that many. So in the end, we ended up with 25 data sets for total P and 8 sites that had full fractionations done. Most of the fractionations we recovered were done using some kind of modified Headley method. I don't know how familiar people are with this stuff, but basically just expose your soil sample to harsher and harsher reagents, and then analyze what comes out in each wash, and then you kind of determine the phosphorus content of the different fractions. Excuse me. we were lucky to have Ann Cross there as part of our group. She basically wrote the review of the method when I was still a little kid. She's got like the, 3,000 citations or whatever on the Headley method. Yeah. And I should also mention that, Headley fractionations are probably the best we have right now as far as a standardized protocol. The residence times of these things can vary dramatically across soils. This is from a recent paper that looked at 53 different soils. Note the log scale on the y-axis. What are you going to do? So that said, as you can see, they do tend to increase with each step, right? The labile P, which is bicarbonate, right? It's faster than the NaOH pool, which is faster than the HCl pool. So we also, while we were at NC's, came up with this fancy conceptual diagram. So these would be 3 locations in a generic ecosystem that vary in their slowly cycling P supply. So here N is kind of shown as the blue gumballs and P is shown as the red gumballs. And the boxes would be the N and P content in the actively cycling soil pool. So the ratio is maintained here for simplicity. So external input and inputs are the same at all these locations. Whatever deposition and fixation are the same. And those are represented by these arrows up top. And so again, where P supply from the slow pool, which is at the bottom, can kind of match N inputs. So the one farthest to the right, and N losses are small. The N loss arrow is pretty tiny, and the concentrations of N and P kind of build up higher in the actively cycling pool. So again, if you were to put this in graphical form, it would look like our hypotheses. So the question is, do we see it? Across sites, we definitely do. So this is total P versus total nitrogen across all the sites in the data set. So higher phosphorus soils do tend to have higher nitrogen. But our conceptual model is actually more about local variation, like we made the cross-site thing because someone's gonna ask for it. So do we see the pattern within site is, I think, the more interesting question. And the answer is yeah, often we do for total P and total N. We found evidence of a spatial correlation between N and P at 16 of the 25 sites. So in each of these figures, the different colors represent different spatial clusters, so like a block. The small points are individual soil cores or pits, and the larger points are the mean for that block. So the black line is just the pseudo-replicated regression of the cores, the individual cores, and the blue lines, when possible, are the regressions of block means. So next, I'll show you the relationships with slowly cycling P for the sites where we had fractionation data. And actually, this looks better than the total P for some reason. So here we're calling slow P the HCl fraction from the Headley. So that's like the apatite I was talking about earlier. In theory, it's primary mineral phosphorus. And so we found some evidence of a correlation in 7 of the 8 sites we looked at between slow P and nitrogen. And the only one where we didn't see it was Tapajos, which is in Brazil. And that's kind of an outlier for a lot of reasons, but it's interesting. So I put it up there. So this is interesting too, because these are drastically different ecosystems with drastically different soil ages, but they're showing the same trend, right? It's a relatively small sample size, but it's North America represented. We have Alaska, New Mexico, New Hampshire, Puerto Rico. And slow P concentrations, like they also vary by orders of magnitude across these sites, right? Like if you look at Coweta and then you look at Hubbard Brook, the they're fairly similar. They're comparable forests. The species composition is different, but they're Appalachian forests. And, soil nitrogen is fairly similar, but the slow P at Hubbard Brook, which is recently glaciated, is like orders of magnitude more than Coweta, where the soils are super weathered, right? But we still see the same trend with nitrogen. So the pattern appears pretty consistent. It's probably fairly safe to assume that within a site, the variability in this apatite reflects very long-term patterns of phosphorus availability. Yeah, I guess I should mention that this apatite pool is within a sample well correlated with the other fractions and total phosphorus, both in this data set and like there's been like global meta-analyses on that. So yeah, I guess just again to illustrate the diversity of the landscapes and break up the graphs. Here's pictures from some of the sites. They're pretty different. So to recap, total soil N and P are spatially correlated within and across sites. The relationship is present even when we look at very slowly cycling P pools, like removing organic matter. And spatial variability in phosphorus ultimately drives spatial patterns of N accumulation and or retention is, I guess, what I'm pushing. So bringing it back to that big picture, we know that the future of nutrient availability is going to be critical to predicting the future of the carbon cycle. The N cycle is more open than the P cycle in most terrestrial systems, and so it tends to adjust to the P cycle because the two things are coupled by biology. So that suggests that we need to be thinking about slower cycling fractions of phosphorus if we want to know how P is going to limit future N stocks and potentially productivity. And I think I have plenty of time left, but thank you. I want to thank my collaborators again. And
Question, Craig. You had on that multi-panel graph, you had one site that looked to be anti-correlated.
yeah.
And I was wondering what was happening there.
Yeah. So I guess the owl's off. So Tim was asking on the multi-site panel for total P, there's one, it's NIWAT 3 in the lower right, where the regression is negative. And so that is a methodological thing, we think. So Dan Lipson is the PI there who gave us that data. And they did not, that's the one site that they generally, their protocol, they don't separate organic and inorganic layers when they're sampling because there isn't a lot of an O. But so I think that's actually just a function of organic matter. And because, yeah, I mean, it's positive in the and NIWAT 1 right next to it. And those are like, close by the two sites.
Craig, this is Satish Gupta. I got some questions for you.
Sure. 1 second. I'm trying to get your, trying to turn the volume up. Go ahead.
So what you're saying is that if you have a slow P, which is a apatite or calcium phosphate, you have a better correlation in terms of nitrogen uptake than if you have a total P. Now, is that mainly because you have some vegetation, which is a long-term vegetation? If you take a, annual crops, would you find the same thing? And the second question is, basically you're saying is if we put an apatite in the soil profile, we can cut down nitrogen leaching.
So I think I'll go with the first question first, which was, oh God, what was it?
Is it really related to the vegetation that you have, which is a long-term?
Oh, is it related to the vegetation?
Yeah. And
you're
not
asking about the
annual...
difference between total P and then the appetite and why the correlation is better with appetite?
Yeah, you got a better correlation with the appetite or slow moving rather than an iron or an aluminum phosphorus.
Yeah. I don't have a great answer for that, but it is interesting. I guess one thing I will say is just based on mass balance, a lot of the when we're showing the correlations with appetite, I'm not trying to like say that the new phosphorus is all coming from appetite. It's a lot of it's coming from like the NaOH fraction, right? Or the
right.
That pool is much larger, and you do generally see the same correlations with that as well. But as to why the correlation seems to work better with the appetite than the total P pool, I have no idea. It's really weird. I mean, it's cool, but
Yeah, I mean, has anybody looked at the mechanism other than just say the trends that you guys are getting are these trends? When you say slow moving, slow phosphorus and showing good correlation, and you guys are excited that, okay, it's picking up a lot more nitrogen and carbon. Has anybody looked at the mechanism, what's causing it? Because what you're saying is you dump a lot of apatite in the soil profile and you can cut down nitrogen leaching and most of the time we know that the plants take up the labile phosphorus or a iron or aluminum phosphorus first before you get a calcium phosphorus.
right. Well, they do, but then but that increases over time as the CNN increase.
Right,
That
it
labile
does.
fraction is what increases. So I think that I guess it's probably better to not focus on the exact fraction of slow. I mean, like the slow P thing is to demonstrate that like the correlation's there even without organic matter. But as far as what specific P fractions are coming or are what's entering or increasing that actively cycling pool, I think that's going to vary dramatically across systems and with a lot of other factors.
So when you say that ignore the correlation, then what's the idea of showing that's your hypothesis that there's a correlation and it says N and P pools go together and increases the productivity. Now you are saying that's not what your hypothesis is. Am I understanding right?
No, I guess the point we're trying to make is that a lot of these, especially in ecosystem science, a lot of folks, they take a malic or they take a bicarbonate extraction and they call that P availability. And over time, that's going to increase with CNN. And we need to be thinking about where the phosphorus is coming from to constrain that increase. And so it
So,
should be slower cycling pools. It's likely the NaO. It's stuff that's moving on us. It has a longer residence time than your labile C or your labile phosphorus. But does that make
well,
sense?
that was my first question then. Are you looking at a long-term vegetation that has roots that are going deeper and maybe getting availability of slow peak?
Yeah,
Anyway,
yeah. He's asking about the vegetation.
maybe I've
Yeah, I
taken
mean,
too much time.
unfortunately, to standardize it, these correlations are all with the top 10 to 15 centimeters. Although at Coweta we had it to 60 and you see and at Hubbard Brook, you see similar correlations with the deeper stuff. But even in those forests, the majority of roots are in the top 30 centimeters.
Hmm. Thanks.
But yeah,
I'm going to
sorry,
let
it was really hard
somebody
to
else
hear
ask
you
it.
because of there's some AV issues.
Any questions for Craig? Craig, what's the main limit on the slow pool of phosphorus availability? So,
I mean, big picture across sites, it's probably water.
yeah.
Sorry,
So
could you repeat the question?
For intensification of the hydrolytic cycle, would you predict them more phosphorus availability?
I would, yeah. I mean, and then also with increasing CO2, I mean, like the rooting, like if there's more roots, like their mycorrhizal fungi can weather it, and um, just the presence of life down there makes more carbonic acid and it speeds up the weathering too.
So would you say then that forest productivity should rise?
Yeah, I mean, well, that's sort of that's what we saw at Coweta, which is a temperate rainforest, so it's not water limited. I mean, it is water limited, but not in the way that, we are here.
A lot of things that are promoting forest productivity, that higher CO2, nitrogen deposition,
And possibly climate, right?
Right.
And so that's sort of what that Rastetter paper was looking at. Like those were the main factors that they were trying to predict. And so, yeah, I guess that's sort of the take home. My take home is like, it's these soil derived nutrient. I mean, calcium, base cations in some places are probably going to be super important. But if you have all these other environmental factors that are releasing limitation from other resources, then the plants are going to need stuff that's in the ground.
Okay, well,
Cool.
thanks so much, Craig.
Yeah, thank you. Oh, no, it's okay. Thank you, everybody.
Out there that you need to check out.
Oh, good call. Yeah.
Some tips.
Animox.
Yeah, very nice. I guess, were you hearing the echo a little bit?
I wasn't too bad, but was it?
It's a little more prevalent back there.
Oh,
but,
man.
yeah, maybe you handled that, just
Yeah.
fine.
Yeah,
Sorry
could you
about
guys
that.
hear what he was saying back there?
Just faintly. I think if you're
I
under
felt-
the speaker,
I felt like I had to like.
yeah. Yes, he's very interested in the phosphorus part.